Yeah, hi everybody. Assuming I can hear this. Here we are. So uh, I'm just basically sending you it's a narration, and hopefully it's from the reproductive system, particularly the female, of some of that material I didn't cover, including the pregnancy and embryology part. That way you can address questions because I'm doing this on Sunday the 23rd. So you'll be able to address any questions that you have with Dr. Hilwig and myself tomorrow. Uh, because I know we have stuff to do before the test that we're going to probably be giving on Wednesday. Should be the 26th. Okay. So the female repro, we went over it. Okay. So basically, I think that the part that I wanted to point out was what brings about that luteal surge, which causes ovulation. That follicle is growing and it's basically being triggered, okay, stimulated by luteinizing hormone, and eventually that's estrogen based, which is causing the follicle to enlarge. Just before ovulation, those levels of the active part of estrogen called estradiol rise rapidly and increase the amount of luteinizing hormone secretions called the luteal surge. Normally, it inhibits it. So it works reverse. Again, this is like a positive feedback. One of those interesting positive feedback things we see in repro, as well as the birth canal and the classic one that's associated with oxytocin. The surge is a positive feedback loop. The estrogen levels, which should be suppressing the luteinizing hormone, actually cause the surge that leads to the follicle bursting out and effectively, eventually, optimally being swept in by the fimbriae into the tubes, and then we covered everything else that was there. Was left as the corpus luteum. The corpus luteum takes over for a better part of about 10 to 14 days, about 10 days actively until, in the absence of implantation, becomes the corpus albicans and effectively triggers menstruation. So the two phases, and these are important, the follicular phase, which begins with the start right after menstruation, right with the development of the follicle, and effectively it even starts during menstruation if you want to get technical as to when it actually that follicle actually starts to grow, and then the luteal phase immediately post ovulation. Okay, so the uterine cycle, typically what they call day zero. Day zero is the onset of menstruation, if you want to think of it this way. So as the follicle matures and begins to release more estradiol, menstruation stops. Then the uterine wall begins to proliferate. So effectively, the estrogen has a double role in causing menstruation to stop over, in that whatever that 3, 4, or 5 day span, depending on the individual, and of course, then leading to the proliferation at that point, or the start of the proliferation of the as well as causing the follicle to grow, proliferating the uterine wall itself. The building phase is what they refer to it as. And basically, that's it. As the corpus luteum is going to take over after the luteal surge, it's going to induce the secretion of much, much larger amounts of progesterone. They're going to really increase and make the wall viable for implantation. If it doesn't occur, then we're going to go through the whole cycle again. Pretty much straightforward. Let's look at what happens when it does occur. And I go over these events toward the end of the term. I wanted to read the ones that they have here, and because I couldn't get a good copy out of the book, although I will try. Okay. I'll try to get most of this in. So, assuming for pregnancy, okay, the cilia sweep it. Fertilization normally takes place in the tubes. Effectively, if you look at the anatomy of the sperm, the enzymes are in the acrosome. And when they contact the egg, the outer area of the egg, they begin to start to digest a path through those granulosa cells and what's called, if you go back and look, what's called the zona pellucida. That's the remnants of, pardon me, the external enclosure around the egg. And effectively, you're going to take multiple sperm, as you will probably already know, to be able to find a pathway for the eventual sperm to be able to enter. So the cell membranes of the sperm head and the oocyte will fuse once there's been a sufficient amount of breakdown. So it's not the first one that does it. and the sperm nucleus enters the cytoplasm of the oocyte. At that point, it's a very important reaction. It's called a cortical reaction, which prevents other sperm from fertilizing the egg. Effectively, that stops the process. Once that sperm, okay, nucleus has entered into the cytoplasm, this reaction hardens the boundary, however you want to express it. But just think of it and obviously there are times when you get more than one, simultaneous things. I mean, they're rare, but that as well as things like non-disjunction can account for some of these polyploidy type of events. Okay, now the oocyte goes through its complete meiosis after fertilization to become an ovum, and there's a second polar body which deteriorates. And effectively, what survives, not the second polar body, but now is the fertilized egg, which effectively becomes the zygote. And then we go through these stages. Now, meiosis is completed. Now we're going to start mitosis and the phases of cleavage, beginning when the zygote is still in the fallopian tube. And it goes through a number of cycles of mitosis. And when it's roughly about 16, this is another number to remember, 16, maybe even 32 cells, is called a morula. The morula is really the first identifiable stage after the zygote stage, and then these multiple cleavages that goes through. The embryo at this stage does not grow during cleavage. Okay. Those cells are what they call equivalent in size and shape and are said to be totipotent, an important word. Totipotent. When you hear the terms about stem cells, these are optimally the ultimate, if you want to think of it this way, totipotent, multiple potential stem cells, something that could theoretically, as science increases, whatever you're feeling about this from an ethical standpoint, is what they're trying to be able to grow organs, to grow a new nervous system, whatever. Okay, they have the potential to express any of their genes. Any one of these 8 cells at this stage could produce another individual. The cells continue to divide for 4 days, forming now this ball becomes hollow with fluid. So it goes from being just a unit of these 16 to 30 or 32 cells. And all of a sudden, you're going to have a cell mass on one side. And you'll see the illustrations where I get this, get better images for you, so to speak. The fluid is called, the fluid filled ball is called the blastocyst. The blastocyst is what lodges in the uterus in a process called implantation about the 7th day after fertilization. The outer cells of the blastocyst implant in the uterine wall and fuse with it, and eventually that's going to be the placenta. You're going to see the mass of cells on one side, and that really becomes significant. Okay, that mass of cells on the inside of the blastocyst closest to the wall are going to become the embryo. And so this is, and all of those, the inner cell mass, so to speak, of this, are the stem cells. These are, when they're harvesting stem cells, this is the kind of thing that they're also looking at. And so we call them pluripotent, meaning they have the ability to develop into most types of cells in the human body, depending on position of the mass. Upon implantation, the female is then said to be pregnant. So understand, totipotent effectively is a cell that could become another individual. Well, pluripotent, now we're looking at something that has the ability to develop into most of the different cells in the human body. So what happens after implantation? The placenta, and it really is not the formed placenta, it's what's going to become the placenta, immediately starts to secrete a very famous peptide called HCG, human chorionic gonadotropin. And that's what's going to maintain the corpus luteum and continue it secreting estrogen and progesterone, and basically maintaining the environment sufficiently for this implanted mass of cells eventually become an embryo and then eventually become a fetus. So the HCG is rapidly detectable. That's the peptide. That's the source of the extremely reliable, pregnancy test. You can purchase for $1.25 at the $1.25 store. So HCG is in the blood and the urine of the mother is the first outward sign. The placenta reaches full development by the end of the first trimester, and then it begins to secrete its own estrogen and progesterone. We don't see the HCG. Okay. So the HCG is what's impacting the corpus luteum, and the corpus luteum will remain active and not turn into the corpus albicans as long, effectively until the end of the first trimester, and that's approximately when the placenta takes over. So the embryo develops past the 8-cell stage. The cells differ from each other. Now, there certainly continues to be communication. The process where a cell is committed to a specialized developmental path we call determination. Okay. And at that point, they're going to start to differentiate into the specialized kinds of cell, whether they're neurological tissue, whether they're organ tissues, whatever. Process is very complex. Okay. It's, they call it gene regulation and development. I'm just reading to you because it has been a very long time, and I'm sure our knowledge of it is much greater. I took embryo in 1973. So that's why I wish I could convey more to you, but I cannot. Okay. Well, there are some things that you do need to be familiar with. Okay. So not all of the cells are terminally differentiated. Skin, liver, and blood cells continue to have stem cells that can regenerate these systems as needed. And we have seen this. When we go over the blood side of the blood, we're going to see that effectively there's a stem cell that produces all the different types of blood cells that continuously is there. The liver continually replaces itself. The skin, you saw that earlier in anatomy when you the skin. Effectively, that's a stem cell. That's that stratum basal. So, and then the researchers are focused on the ability to generate these kinds of stem cells from other kinds of stem cells so that we have regeneration. The formation of what's called the gastrula, okay, and it's a process called gastrulation, occurs about the third week after fertilization. So at this point, the cell movements occur as the cell begins to slowly move about the embryo. In mammals, they form something called a primitive streak, and you'll see these in the images, if I can get them done properly. If not, I'll just try to take a really good camera shot and send them to you, which is analogous to the blastospore in aquatic vertebrates, if that was something you're familiar with from zoo. Cells destined to become mesoderm migrate into the primitive streak, and during gastrulation, we form the three primary germ layers. You must know the germ layers. Okay. Ectoderm, mesoderm, and endoderm. Ectoderm is skin, it's outside, ecto. Endo, inside. Meso, in the middle. Meso is mostly all the connective tissue. So and I'll give you the specifics. The ectoderm, not only is it skin, but it also forms what's called the neural streak, which is the start of the nervous system. And if I didn't tell you already, understand that a lot of diseases and conditions, and particularly hereditary conditions that affect the nervous system. I'm not talking about depression, or I'm not talking about schizophrenia. I'm talking about things that have psychiatric, psychological implications. But most famously, neurofibromatosis. Typically, we see in something called tuberous sclerosis, are diseases where there are characteristic skin lesions that facilitate the diagnosis. In neurofibromatosis, which in one of its manifestations is the elephant man, these individuals have what are called cafe au lait spots. They are just that kind of a color, like a brown to a pale brown color, that typically increase around the time of puberty, and typically are abdominally or approaching the area of the genitalia where they predominate, that are there. So, I mean, this is, and you see this. So we look for characteristic ones. Those folks, because my first wife has this, and it's an autosomal dominant disorder. I'm very familiar with it because I'm familiar with her family and all the folks that have had this and problems attended to it. I mean, things like strawberry hemangioma. It's a freckle, but it's a specific looking kind of freckle. So here's what they tell you: the epidermis, the ectoderm, epidermis of skin, nervous system, and the sense organs. So you're looking at eyes and the hearing apparatus, taste, smell. The mesoderm, think connective tissue, middle. Okay, bones, muscles, blood vessels, the heart. Now, some of the organs are not that, but the heart basically is something that's mostly made of different kinds of connective tissue. So skeleton, muscles, blood vessels, heart, the blood itself, gonads, interestingly enough, kidneys, which is mostly blood vessels, and tubes, and dermis of the skin. That's clearly a connective tissue. When we look at the lining of the digestive and the respiratory tracts, the liver, pancreas, thymus, and thyroid, they're endoderm. So to me, endoderm, if I was going to paint with a broad brush, okay, I'm thinking more of digestive organs, respiratory. I'm thinking of, my liver and pancreas are all a part of digestive. The thymus and thyroid, which things you might just need to remember. It's the best I can tell you about that. So there's no absolute rules for memorizing these. Okay, certain guidelines to be followed for the MCAT. The ectodermal cells, outer covering, skin, nails, teeth, tooth enamel, nerves, nervous system, sense organs, endodermal cells, lining of the digestive tract, much of the liver and pancreas, mesoderm, into the structures that lie in between the inner and outer coverings of the body. So if that's the default, so skin and nervous system, easy for ectoderm. And then you just have to think of internal linings for digestive and respiratory. Remember, liver and pancreas, and pretty much everything else is mesoderm. So in the third week, the gastrula develops into the neurula in a process called neurulation, and you begin to find those things that you remember, I'm sure, from zoo and related even high school biology. The notochord from mesoderm, okay, and it forms something called the neural plate. The notochord deteriorates, neural tube forms from the neural plate, become the spinal cord, brain, and most of the nervous system. The cells of the ectoderm that are close to the neural tube are known as the neural crest. The cells of the neural crest, mostly accessory cells such as Schwann cells. So I mean, all of this, and again, I'll rather than just me read it, but you understand all of those things. And these neural crest cells will migrate to different areas through embryological development. And so we understand the role that apoptosis plays in a lot of these things, uh, even for parts of the nervous system. Toes are webbed. If you did not, fingers and toes are webbed. Those go away. Okay, and again, when apoptosis fails, then we have cells that grow and sometimes grow irregularly, and that's where cancer is. And it's such really an important process. And then when we stop, then the end stage of this is senescence. So we do something at the very end of phys when we start reviewing growth and development and senescence. And I think that's really what you need to know about it. Okay, when they talk about prenatal development, the development that occurs before birth, development of the embryo, following the early stages of development, etc., etc. By the 9th week of pregnancy, major organs are in their final locations. Developing organism is now considered to be a fetus. So, roughly, okay, depending on how you would think, 9th or 10th week is probably the transition from the embryo. Okay, so the embryo would be in the early stages when we begin to identify. Maybe in a couple of weeks, you can look up to see what is accepted. 9th week of pregnancy, we're looking at, effectively, we call it a fetus. Some people are looking at even for by the end of the first trimester, that would be 13 weeks. So, I mean, I would think you would look up to see the current numbers that are associated when we make that transmission. 2nd trimester, but the fetus is not yet capable of independent survival. The 3rd trimester, because of the development of the respiratory system and the ability to form something we've covered in respiratory and will cover again in respiratory, the development of surfactant to allow the lungs to expand and contract. I mean, at that point, now we could, even a preemie. Years ago, it was 26 weeks was the number. If it was anything before that, the odds were very unlikely to be able to survive a premature birth. 24, now it's 24 to 26, even younger, sometimes 23, depending on circumstances, with our ability to effectively manufacture the surfactant and keep this extraordinarily tiny, neonate to be able to survive. Okay, the brain's not certainly developed that certainly at the time of parturition or birth. Okay, it just wouldn't pass its way through there. So I mean, you have to understand that because you have to look at what's normal. Normal is a vaginal birth, and so the issues of development came immediately postpartum. We begin to see these things. That's why all of those wonderful landmarks, when a baby can hold her head up and roll over and sit up and certainly walk, etc., etc., is also interesting, significant, and there's a fair amount of difference between them. The watchword here is called cephalocaudal, from sort of like from head to tail, and for our purposes, that's why head control is certainly this thing that we know is roughly between 3 and 6 months, and the average time plus or minus probably at least 3 months, maybe four, for walking. 12 to 15 months is the classic time for this. So, I mean, all of those types of things are things you should look at, and pretty much that's what I have to say to conclude this chapter. Hopefully, we'll get a chance to look at the questions, the 8 or so questions that there were at the end of this unit, and thank you very much. That is all.